Dear learners, welcome to CCE LMS portal. Myself Sriranjani Tallam, lecturer in zoology, DK Government College for Women Autonomous Nellore. In this video lesson today we are learning about the topic diseases of shellfishes, which comes under the syllabus of 5th semester Aquaculture Technology. Let us see about the learning outcomes of this lesson. Upon the completion of this diseases shellfishes, students will learn about diseases introduction, what are diseases, what is their definition according to scientists, and he will learn about viral diseases diagnosis and prevention, and then students will understand and learn about different bacterial diseases occurring in shellfishes, their diagnosis and prevention. Finally, students will learn about protozoan diseases, diagnosis and prevention. Let us start with the introduction to diseases of shellfishes. The global aquaculture activity has been recognized as the fastest growing enterprise with an estimated annual growth of 11%. Globally, shrimp continues to be largest single seafood community by value, accounting for 15% of all internationally traded fishery products. India is blessed with a long coastline of over 8,000 kilometers, providing immense scope for both brackish water and freshwater aquaculture. Aquaculture activity in India has been contributing significantly in the various spheres of people's development in terms of providing livelihood, food security, employment, and trade. Farm raised shrimp is comprised of 55% of global shrimp production, and this is entirely dominated by two species, black tiger shrimp Peneus monodon and Pacific white shrimp Peneus vename, according to Food and Agriculture Organization 2012 statistics. However, increasing trend in the intensification and commercialization of aquatic industry resulted in the increased disease outbreak. WSSV white spot syndrome virus alone is causing annual loss to the tune of 300 crore rupees since 1994 to the shrimp culture industry. Let us learn about definition of disease. Disease outbreak is an end result of negative interaction between pathogen, host and environment. We can see in this image pathogen, host and environment in the negative interaction will finally lead to formation of a disease. So this is host, this is environment and this is pathogen. These three interact in a negative way so that it will result in the disease according to Sijko 1974. Disease is now a primary constraint to the culture of many aquatic species, impeding both economical and social development in many countries. Shellfish diseases. There are three types of shellfish diseases occurring in shellfishes: viral diseases, bacterial diseases, and protozoan diseases. In this video lesson, we will learn in detail about viral, bacterial, and protozoan diseases in the case of shrimp. Let us learn about viral diseases, prevention, and therapy. The first and foremost viral diseases we are learning about is baculovirus midgut disease. The causative agent for this disease is baculovirus midgut gland necrosis virus. In short, we can call BMNV, a non-occluded gut infecting baculovirus. Non-occluded means not stopping or not blocking gut infecting baculovirus. It is also called as midgut gland cloudy disease, white turbid liver disease. The species affected by this diseases are Peneus japanicus and Peneus monodon. Usually, this virus affects larva and early post-larval stages of the penis. And the gross signs are sudden onset of white turbid midgut gland hepatopancreas in larvae and post-larvae with associated high mortality. White turbidity of hepatopancreas is clearly observed. In this image, we can see the hepatopancreas is white and turbid. This is the primary gross sign of the baculovirus midgut disease. Effect on the host. Generally, this virus infects larvae and early post-larval stages in which it can cause high mortalities. The apparent white turbidity of hepatopancreas is caused by necrosis of hepatopancreas tubule epithelium and possibly also the mucal epithelium. Larvae float inactively, but later stages, that is late post-larvae, tend to show resistance to the disease. We can clearly see white turbid midgut line through the abdomen. The sources of diseases are through wild caught female spanners, and we can diagnose this disease through appearance of necrotic hepatopancreas midgut gland tubule, and epithelial cells show nuclear hypertrophy, that is increase in size, and chromatin margination. The chromatin material moves towards the margin of the nuclear membrane with marginated chromatin. Preventive measures or methods of control. No known treatment is available right now to this disease. We have to destroy the infected stocks and disinfect equipment and tanks which are used in these ponds affected with this virus. And we can avoid use of baculovirus midgut necrosis infected brood stock completely. And a prophylactic effect can be achieved by washing fertilized eggs in the clean seawater. By following these preventive measures, we can prevent the occurrence of this baculovirus midgut necrosis viral disease. Students, let us learn about very important virus, white spot syndrome virus, which causes severe loss to aquaculture industry. This white spot syndrome virus causes white spot disease in organisms, that is particularly shellfishes. The causative agent is large double-stranded DNA virus, belongs to genus Vispovirus of family Nemaviridae according to International Committee of Taxonomy of Viruses, that is ICTV. They have placed these viruses in a new genus called Vispovirus under the family Nemaviridae based on its unique morphology and genomic composition. The species affected by this WSSV are: WSSV can affect a wide range of aquatic crustaceans, including marine, brackish, and freshwater peanuts, crabs, and crayfish. So, this virus can transmit both horizontal transmission and also vertical transmission. Vertical transmission from one generation to other generation, from parents to offsprings. Horizontal transmission is from horizontal way through waterborne or foodborne or from organism to organism, that is called horizontal transmissions. Gross signs of this disease. Affected shrimps are lethargic, show reddish body discoloration, and during advanced stage of disease, characteristic 1 to 2 mm diameter white spots could be seen on carapace, appendages, and inside surfaces. So this virus severely damages shrimp stomach, gills, heart, and eyes. Cumulative mortality typically reaches 100% within 3 to 7 days of infection. Effects on host. There will be abrupt reduction in food intake, lethargy, anorexia, and loose cuticle. We can observe calcified deposits on carapace which ranges from 1 to 2 mm to nearly 1 centimeter. We can see in this image the former is showing carapace with the white spots. Affected shrimp comes to burn and dies. Cumulative mortality is 100% within 3 to 7 days of infection causing huge loss in the aquaculture industry. We can diagnose this WSD, white spot disease, can be diagnosed based on the gross signs such as presence of gastric white spots and rapid mortalities. And we can histopathologically detect this disease, which is a gold coin in the case of detection. We can confirm this white spot virus through PCR or molecular tools such as in situ hybridization tests. In this picture, we can clearly see the target organs for WSSV disease: cuticular epithelium, hepatopoietic tissue, antennal gland, stomach epithelium, and gills are all affected with this WSSV disease. Students, let us learn about preventive measures of WSSV. As there is no permanent treatment for WSSV, the farmer has to follow certain preventive measures so as to avoid the occurrence of white spot disease. Drying the pond between the crops is the first preventive measure. So between the two crops, the pond should be completely dried and lime should be applied so to erase the viruses which is present in the soil also. Virus can transmit through carriers like rotifers, artemia, molluscans. So the farmer has to sanitize and filter the pond water before stocking. The farmer has to stock the PCR, polymerase chain reaction tested specific pathogen free SPF quality seed so as to avoid the occurrence of WSSV. They have to follow good management practices and strict biosecurity measures so as to avoid the entry of disease in the culture. Washing and disinfection of eggs and apple should be done to avoid the WSSV. Here we can see the Venami showing the white spots. So white spot syndrome virus can be avoided by following certain preventive measures. Students, let us learn and understand about infectious hypodermal hematopoietic necrosis IHHN virus and disease and pathogenicity and preventive measures. Infectious hypodermal and hematopoietic necrosis IHHN was first discovered in Penis venae and Penis stylirostris in America in the year 1981. IHHN is caused by small single-stranded ranging from 20 to 22 nanometers parvovirus. It can transmit both horizontally and vertically. And the species affected are Penis venae and Penis stylirostris. Gross signs are The gross signs of disease are not specific to IHHN but may include reduced feeding, elevated morbidity and mortality rates, fouling by epicommensals, and bluish body coloration. IHHN can cause runt deformity syndrome and deformed cuticle and rostrum. We can see in this picture the deformed rostrum in the L. venae. Effects on the host. Reduction in feed consumption, changes in behavior and appearance. In Penis venae, these viruses IHHN cause runt deformity syndrome resulting in bent rostrum, slow growth and poor feed conversion ratio. As a result, harvest is lowered and there will be a reduction in profits. So we can see in this image the venae, the deformed rostrum abnormality in walking legs can be observed. So preventive measures. No effective vaccination methods for IHHNV has been developed. Farmers should stock PCR tested quality seed. Ponds must be completely dried in between two crops. We have to follow strict biosecurity measures and we have to follow good management practices. In this image we can see the ponds are being covered, strict biosecurity measures are being followed. In this way we can prevent the IHHN virus. The next disease we are learning about is yellowhead disease. The causative agent is rod-shaped enveloped virus of 40 to 60 nanometers by 150 to 200 nanometer size containing single strand RNA, the yellowhead virus. Yellowhead virus principally affected pond red juvenile stages of 5 to 15 grams size. And the species affected are Peneus monodon and Peneus vename. The gross signs include cephalothorax become yellowish. We can see in this picture the Peneus monodon affecting the cephalothorax has become yellowish in color. The disease is transmitted exclusively through horizontal transmission. We can observe swollen and light colored hepatopancreas, and the overall shrimp appearance is pale in color. So let us learn about the effect on host. This yellowhead virus affected shrimp will feed voraciously for 2 to 3 days and stops feeding, and we can observe them on pond periphery. This virus affects tissues of ectodermal and mesodermal origin, that is, it affects lymphoid organs, hematocytes, gill lamellae, spongy tissues of gut, gonads, and nerve ganglia. We can diagnose the yellowhead disease through using RT-PCR and in situ hybridization test. We can also observe histological basophilic inclusion bodies in the case of stomach and gill. So the preventive measures which should be followed are farmers should stock PCR tested quality seed, pond must be completely dried between two crops and strict biosecurity measures, and we should follow good management practices. YHD eradication in ponds is similar to that of other viruses and involves practicing best management practices. Students, let us learn about bacterial diseases, prevention and therapy in the shrimp. So let us start with a very important disease, vibriosis, causing huge loss to the shrimp industry. The vibriosis is severe systemic affecting whole body bacterial disease. Two types of two species of gram-negative Vibrio species of bacteria, Vibrio harvae and Vibrio parahemolyticus, are responsible for this vibriosis. This vibriosis easily affects shrimp culture in saline waters, causing bacterial septicemia. It can affect any stage of the shrimp. The environmental stresses aggravate this vibriosis, causing huge loss to aquaculture industry. We can see the bacterial septicemia vibriosis in this picture. We can observe this is the healthy shrimp and this is the sick shrimp due to vibriosis. Symptoms of vibriosis. The shrimps affected with the vibriosis are lethargic. They exhibit abnormal swimming with loss of appetite and loss of reflex and cuticular fouling. Gills appear brown and red. Atrophied hepatopancreas. We can see here discolored hepatopancreas in this image. Affected shrimp does not eat, that is, loss of appetite can be observed. Blackening of basal part of antenna and mortality in serious condition of vibriosis can be observed. Vibriosis leads to tail rot in the case of shrimp. Symptoms are muscle tissue of tail with the reddish and decay, burn like spots entire the body, loss of body parts, lethargic movement, abnormal swimming. We can here clearly see the tail rot condition. So the tail is rotted in the case of this shrimp. So preventive measures and control of vibriosis. Drying of pond after the production cycle, following strict biosecurity measures, using good quality water and bacterial free seed selection, medicated feeds like oxytetracycline 1.5 gram per kg fed at 2 to 10% of body weight, and we have to maintain the pH management of water, and probiotics can be used to reduce the vibriosis. These sources of Vibrio harvae in Peneus monodon hatcheries. Spawners release fecal matter in spawning water. Adherence resulted to the biofilm formation on egg surface. This egg surface is having the Vibriosis bacteria. This proves that eggs are colonized by the Vibrio harvae before hatching, and this pathogen route can be disrupted by spanner removal and egg rinsing. So these are the preventive measures and control to follow in the vibriosis condition. Luminous bacterial disease causative agent is Vibrio harvae and Vibrio splendidis. Species affected are Peneus monodon, Peneus merguensis and Peneus indicus. Gross signs are shrimps become weak and opaque white. Affected shrimps often swim to the pond surface and edges. Heavily infected shrimps in tanks and ponds show a continuous green glow. That's why it is called as luminous bacterial disease. So you can in this image Peneus monodon is suffering with the luminous bacterial disease. Effects on host. The hepatopancreas is the target organ of luminous bacteria. The lesions affect the digestive function of the organ as the necrotic parts become non-functional. Total necrosis and disinfection leads to death, while partial disinfection causes slow growth as not all tubules function in digestion, absorption and storage. Diagnosis. This luminous bacterial disease may be detected by bacteriological, histological, serological, and fluorescent antibody technique FAT and ELISA enzyme-linked immunosorbent assay using specific antibody methods. Prevention and control. We have to disinfect the incoming water and use filtration equipment to prevent the entry of luminous bacteria into the hatchery system. We have to use immune prophylaxis or vaccination. The farmer has to monitor bacterial population and diversity in the intake and rearing of waters of the shrimp pond. The farmer can apply antibiotics and other antibacterial substances only as the last resort. Let us learn about filamentous bacterial disease. The causative agents are Leukotrichs species, Thiotrix species, Flexibacteria, Cytophaga, Flavobacterium species. And the species affected are Peneus monodon, Peneus merguensis and Peneus indicus. The gross signs of filamentous bacterial diseases are presence of fine, colorless, thread-like growth on the body surface and gills as seen in the coming images under a microscope. We can see in this image these are the filamentous bacterial diseases causing filaments. Effects on host. Infected egg shows a thick mat of filaments on the surface. In larvae and post-larvae, normal respiration, feeding, locomotion and molting may be seriously impaired, resulting in slower growth rates. Frequent molting does not allow adequate time for the bacteria to accumulate on the exoskeleton. Mortality is due to hypoxia or loss of respiration. Diagnosis. Direct microscopic examination of wet mounds of larvae or post-larvae, appendages and gill filaments excised from juvenile or adult shrimp and filamentous organisms attached to the external surface of cuticle can be seen. Prevention and control. We have to maintain good quality water with optimum dissolved oxygen and low organic matter levels. Apply cetirizine plus at 0.15 ppm copper in 24-hour flow through treatments and apply 0.5 ppm copper in 4 to 6 hour static treatments for post-larval stages 2 and older. Let us learn about shellfish disease or brown black spot disease. Causative agents are Vibrio, Aeromonas and Pseudomonas groups and species affect are Peneus monodon, Peneus merguensis and Peneus indicans. The gross signs shown in this brown black spot diseases are the disease manifests itself as a brownish to black single or multiple eroded areas on general body, cuticle, appendages and gills. In larvae and post-larval stages, the affected appendages shows a cigarette butt-like appearance. So effects on host. Infection usually starts at the sites of puncture or injuries caused by telson or rostrum. The bacteria produce extracellular lipases, proteases and chitinases which together erode the multiple layered cuticle resulting in the development of this brown black brown black disease. So we can clearly see this brown or black spots in these images affecting the shrimp severely. Diagnosis, prevention and control. Disease peanuts are examined for appearance of multifocal melanized cuticular lesions on the cuticle or on the general body surface, appendages or on the gills. Diagnosis may also be made by bacteriological and serological methods. Prevention and control. So earlier we have discussed the same prevention and control measures can be done here also. Protozoan fouling. The protozoan fouling are caused by protozoans namely Verticella, Zoothamnium, Epistylus, Trichodina, Acinata and Ephilota. Symptoms are slimy exoskeleton, reddish to brownish gill, restless condition. Effects on host. Locomotion and respiration is hampered in the case of shrimps. Fuzzy mat-like appearance due to ciliate fouling, and gills are choked causing mortalities in the shrimps. So we can clearly see these are the protozoans which are affecting the shrimps. Trichodina parasite, we can see Verticella and Zoothamnium. In this image we can see the shrimp which is severely affected by the Zoothamnium infestation. So diagnosis. Microscopic protozoans are observed attached to the external parts of the shrimp in microscope. And preventive measures are maintaining good water quality, avoiding heavy organic load, avoiding high nutrient supply and following good management practices. This is the image of Ephilota which is causing a protozoan fouling, and this is the image of Epistylus affecting the protozoan fouling. So, students, through this lesson, we have learnt about different shellfish diseases caused by viruses, bacteria, and protozoans.